really like to thank the organizers for inviting me and giving this opportunity to talk here. I'm going to tell you about a title called Interactome-based drug discovery, really trying to make a case that drug discovery is not an interaction with few targets, but really an iterative process which happens with interaction by taking into account with all biomolecules. And how we do that, I'm going to put an example and show you some wet lab result as well. So this is a story I want to share with you, which started really at Stanford right after my PhD, and in collaboration at the time with University of Washington, when after the PI moved to sunny Buffalo, this approach was developed after my PhD, and then I went on to do some wet lab work and some mouse work, being a computational biologist, and that was fun. It's amazing how much you can learn from YouTube if you've never held a pipette, which I never did. So this problem, Chad really gave a good introduction previously in the session as to what the problem is. And obviously the problem is that the drug pipeline is drying up. And this was a blog post by one of the Eli Lilly's director of communications. And she said that it costed $1.3 billion to bring a new drug to market. And it's even more staggering than that if you take into account the past 15 years of the amount of money which has been spent. But just to put things in perspective, this is actually a lot of happiness. You can see that from an American football perspective, these are 371 Super Bowl ads. You can buy 2 pro football stadiums and every NFL ticket in every stadium for about 6 weeks. So I think a lot of happiness, and I think this is probably the primary motivation. We should reduce drug discovery costs to a great deal. But just to be on a serious note, what we know really about Moore's law, which is a computer science law which everyone knows here, that the cost of purchasing or computing is decreasing, or cost of getting more and more computers is increasing. What I famously term Eroom's law is the law of drug discovery, where the drugs are getting more and more expensive with time. At the time of around 2010, when I finished my PhD, this article came out in The Economist, and it was really talking about the data deluge. And it was interesting that how much data as a species we produce, which is really 2 billion terabytes, which is 2 zettabytes of data and stuff. And the biochemical data, if you see, even this morning, you can see that there are about 1 million assays which are there in Array Express. But I would like to make a case that this big data by itself is not enough. I think it's great. We can combine a lot of data. We can get a lot of things out of it, which many people in the conference and otherwise have shown. But to some extent, biophysical modeling in combination with this would give a much better atomic understanding and perspective of how to do drug discovery. So kind of before even going forward with it, I would like to say that how does biology work? In a sense, in my view, biology is really very redundant. There are so many pathways to do similar things that just choosing 1 or 2 targets as protein targets and doing drug discovery, I think, is inherently flawed in some ways. And I would like to say this even more so, that from a simple computational experiment anyone can do, take all the human proteins or the entire human proteome and try to find similarity of binding sites, active binding sites among the human proteome. Here I'm showing just a part of it. And you can very well guess what this cluster is, which is obviously a kinase cluster where the binding site are conserved. You can immediately see that taking a few targets as drug targets can immediately lead to a lot of toxicity. And really, drugs are failing 95% of the time, mostly because of either pharmacokinetics, efficacy, or toxicity. And these 3 things are very important. So I'm only going to focus on right now a solution which is one of these legs, and hopefully in the future attack the others. So what I'm going to talk about is these 4 things. The solution, what we think can be applied to this, which we infamously call CAN-DO platform, and CAN-DO just stands for computational analysis of novel drug opportunities, and then talk about the benchmarking as to, with this theory, how well does it benchmark with all diseases and all drugs out there. Give you a flavor of one of the application and the wet lab experiment which I did, and talk about the future work and perspective. So, so I think the solution really lies by taking all the protein structures out there and modeling a large portion of many different proteomes, and then using all the human-approved drugs for now, and then finding interactions between them. So when I say kind of all of this data, I mean, obviously there is a lot of modeling here, but it kind of amounts to about 1 billion calculations or more, which is really this part where this is all human-approved drugs, and this is a multi-organism proteome. And this is kind of a signature. In similar ways, what Chad was explaining, a signature of genetic profile, this is really a signature of structural interaction, if you will. But I would also argue that this signature, rather than doing one protein at a time, this is somewhat of a functional signature of a compound. Rather than comparing structures of compounds, it is much better to compare the signature of these compounds. And like I said, these are all human-approved drugs for now. There are 24,000 fragments in total from a few thousand compounds. But what we will notice that the unique fragments which are there is only about 20% of that. And that makes sense, one, because pharma uses a lot of fragments over and over again, but more so, it makes sense if you remember from the figure I showed you about binding sites, that the binding sites are conserved in different regions, and a lot of them are kinase, multi-kinase inhibitors and others which are there in the market. So it's not imperative that there is less number of fragments which actually can reduce toxicity and actually become drugs. So, but this by no means is saying that this is the end. There is a whole lot to be explored, which I think I can mention and so on. The thing which I want to say is what we know about biology and what biology does best is homology. So I'm using the word homology very loosely here and making a hypothesis or a case that if there is a compound which has a particular functional signature, and if there is any other compound with a similar signature, those two compounds would behave in a similar way. So that's what I'm trying to use as a basis of, you could say, repurposing or putting drugs out there. So one of the methods, I would just try to give you a flavor of how we do this in terms of how can you actually do a massive screen like this. Obviously, if you do this one at a time, it'll take a long time, and I did not have that much patience to do because I also wanted to do experimental work. So one of the things which we developed worked on a few tenets, and this is not perfect by any means, but for our case, it worked quite well. And one of the tenets was that binding site is more conserved than sequence and structure. And the other tenet, which I already showed you, that a lot of human-approved drugs uses similar fragments. So let's look at the entire PDB, and in the entire PDB, at a drug whole compound level, we don't get matches. But at a fragment level, there were a lot of matches of fragments with the fragments of the PDB ligands. Along with looking at the microenvironment of these fragments, we can cluster these fragments in the binding pocket of a protein. So you can think of a protein having a binding site, and all the blue points here are the positions of the fragment which occurs of similar binding sites which are there on the entire PDB. And these blue points, and there are some ab initio predictive calculations here also, but mostly I'm showing which are there in all of the PDB at those micro locations. But because you're doing this for each fragment, for your whole compound library, you can actually do this in one shot. And actually for all different fragments shown by different colors here, you can really create where all these fragments can come inside the binding pocket. So in some ways, you can, in one shot, know in a rigid docking way for now, where all these fragments are. You can make your compounds, and you can also design new compounds, synthesize them from a perspective of where a certain carbon-carbon bond is, which can be synthesized by transition metal pyridium reactions and so on, and you can give those predictions. But biology and function is not static. It is dynamic. So to really achieve flexibility and put flexibility into it, we kind of went back into what is very well known in the protein structure prediction field. It is very well known how to do threading in protein structure prediction. It is very well known if you have certain part of structure which is matching or not, how you can take that information and combine and join. So that's exactly what we did in terms of kind of joining these different fragments up from these different locations, at the same time having a flexibility of the protein. So this kind of was quite fast, and the future versions of these methods are done in collaboration with Janez Konk in Slovenia. So this is one of the methods which is used per se, but I hope it gives you a flavor of taking a network of binding sites and how to combine them. And on top of that, I would want to, at a disease-specific level, include the expression data. So it doesn't make sense, let's say, if in a particular tissue where the disease arises and the protein is not there, no matter how much better prediction you get as a binding to that particular compound of a protein, if it's not expressed in the tissue, it's not expressed. It doesn't make sense to even consider that to some extent. So that essentially what it does, it kind of adds in some way color to this interaction matrix. And where a positive and a negative interaction is really a measure of if it's good or bad for the disease in that sense. And again, one can do this at a disease-specific level, get these all versus all interactions, and try to make the case with the same hypothesis that similar networks or similar signatures would do similar things and in certain way. So like I said, this is a basic hypothesis, but it is by no means valid if you cannot retrospectively and prospectively benchmark and test it. So from a benchmarking perspective, what we did, we just took all the diseases out there which had at least 2 FDA-approved compounds, and then we asked the question, if we remove one of those compounds every time and then do and see how well we can retrieve the other drugs which we have removed from this for all 1,400 or so diseases. And what we get is what I'm showing you here is an indication compound pair, that is a compound which is removed for that indication, sorted, and the prediction sorted by a particular measure. In this case, we tested a lot of different metrics, and one of the ones which came up was this RMSD metric with the number of proteins, which gave a good result as far as benchmarking was concerned. So you can have these top 10, top 30, top 50, top 100 compounds which one could test. And really the accuracy of these compounds is the number of compounds approved for an indication at a given rank divided by total number of compounds. So what that really means is, for example, for this particular disease, compound pair, there is only one compound which is predicted in the top 10. So you have a 25% prediction accuracy for that. So, okay, so I think let me just skip over all of these because I think probably my time is getting short. So I would like to say that this is all in the paper also. You can read up that the different protein classes do have an effect, but if you consider all of the protein classes at the same time, then you really get maximal accuracy. That doesn't mean that different protein classes like kinases and GPCRs are not important. I think this tool also gives a perspective of how these different protein classes can be done for different diseases. As an example, for hereditary angiogemas, where it's known nuclear receptor is a part of it, there were 2 other predictions, and biologists can go back and test this. So let me give you a flavor about an application which is there very quickly. And this is type 1 diabetes, and this is the work I did myself in mice and others. And type 1 diabetes is an autoimmune disease and a metabolic disease as well. It occurs in the pancreas, and these big circles are the islets in the pancreas. Inside these big circles are beta cells, which produce insulin. In type 1 diabetes, there are pathogenic T cells which enter these islets and then kill off these beta cells. So the less and less insulin is produced, and with time, it's a devastating disease which gives rise to organ failure and many other things. Without going into detail again, I'll just explain a little bit about how this was done. So to do type 1 diabetes predictions, I looked at all the approved drugs for type 2 diabetes, and one would ask, oh, these are different diseases. But I think there is a common connection of the organ which is there for this disease, and the biology in terms of what happens in glucose control and others is important. So in some ways, I generated the signature with all the type 2 diabetes drugs and asked the question, how many signatures matches each of these in a combined way, that if you combine all these signatures together, what are the compounds which you can retrieve from there to really make an edge between a a compound indication space as a prediction, including some of the pathways which are already known for type 1 diabetes, which I had to explain in the previous slide, but I'll just skip over it. So anyways, we get this interaction map for compounds with proteins, and very naively I selected, let's see the ones which interact with most proteins as the top one, and let's go from there. And again, I can do this right now because these are all FDA-approved drugs or human-approved drugs which I can test in mice. And this was the compound which came up, and I'm just going to show you a data very quickly of NOD, non-obese diabetic mice, which was done for both type 1 diabetes reversal and for prevention. So for reversal, essentially, the mice were NOD female mice which were treated daily for 9 weeks, and they were treated when diabetes started, which was 2 readings of 250 for the glucose, and for 9 weeks they were treated, and this is the result. So what you see in the shaded portion is the drug treatment, and you see that the diabetes incidence was 100%, and within a few days it drops for all 100% of the mice. But it kind of went up to 15 or 10% during the drug treatment out of the 11 mice which were drug and 11 which were vehicle treated. And when you stop the drug at 9 weeks, these mice, only about 40% of them remain diabetic. Rest 60% of them are diabetes free. So in a sense, this is saying that there is some amount of tolerance which is induced possibly in these mice, that even when you stop the drug treatment, there is a way to continue that. And this is also kind of shown here that in an islet, you see for drug treated, there is very little blue points, which are really the pathogenic cells. And in a vehicle treated, I hope you can see there are a lot of blue points which has infiltrated, which kind of kills the mice. Similarly, a similar prevention study was done, which was that at 12 weeks of age, for 9 weeks daily, these mice were treated by gavage, which is by the mouth, and you can see that there is a delay in diabetes, and there is a difference between control and drug treated. Do I have 2 minutes? Okay. So let me say that we have tested this for many other diseases with collaborators, obviously, and we have a lot of different results, which I don't have time to go into, but the whole idea which I want to the audience to bring home to is that it's taken into account this signature of an interaction signature of these compounds, which is really making possible some of these. And by no means, this is a one-man show. Like I said, this is a lot of collaborations, which is there, and it's work of the past 5 years, per se, and I'm happy to talk to anyone. I'm looking for collaborators all the time. So let me just skip over this and come right to the point, because you said there is no time. So the future work is something which we are trying to do to join structural biology with cell biology, to join my experience in computational structural biology and immunology with developing a structural cell signature in the hope, and this could be a science fiction dream, that I could proliferate cell types in vivo, up or down, depending on a disease. For example, for autoimmune disease, I would like to proliferate the regulatory T cells so that the autoimmunity is cured, and for cancer, to down-regulate regulatory T cells with these molecules. So in some way, a dream of developing cell-responsive drugs. And to make this science fiction a reality, I've been offered a job at Purdue University starting in January, and this is an advertisement just for people to see. We have a nice building, and I'm looking for postdocs, and I'll be very happy to work with people much smarter than me. And we have a wet lab, and if you like to play with robots and computers, and in the hope to save some lives, hopefully, please come and join and talk to me, and I'll be very happy to do it. So this is my last slide, I promise, or second to last. So I would like to just thank my mentors, Michael Levitt, my PhD mentor, who was really far-sighted in telling me that after my PhD, just stay for 2 years and do whatever you want to do. And this is what I developed in collaboration with Ram, who really paid for all these experiments and has been an excellent mentor. Roger has been a big supporter of the computational work. And I'm indebted to Jeff Bluestone, who's an immunologist at UCSF, who, me having no experience in experiments, really took me in and had me do this. So one of the most important organisms here is obviously the mice, but without which no work would have been possible. And thank you again, and sorry for the time. Thank you for listening.
We can take a quick question.
I don't think there is.
Yeah,
Oh,
so is there a question? Sometimes it's hard to see in this
yeah,
room.
there's one person coming.
Okay. Yeah. Actually,
Yeah.
I think best to take it offline. So I would like to thank Gaurav and the three speakers for an excellent session. Thank you very much.